minutes. So I'll just get started. So it's going to cover everything from what we do in the lab in genomics to everything we've seen in the subway and a little bit of work with him looking at NASA. So I'll start quickly with an intro. And actually, can everyone hear me okay if I talk like this, I suppose? Okay, so as most of you know, the molecular recipe for the synthesis of all the cells in our body starts out as one cell, and everything's structured with it to become all the cells in your body, whether you say an cnidarian or an invertebrate or a vertebrate looking at other invertebrates, or in the case of developing your own progression, molecular progression like my daughter who ends up liking to love ice cream, I shamelessly use her. She has approved this picture that I can use her. But in all cases, the instructions are present at day one. That's actually part of the reason I became a geneticist. I'm still fascinated to this day by that simple fact. And so the question is exactly how does that happen? So we know that this is an interplay between DNA and RNA and also proteins coming together to create what is arguably an understatement to call complex behavior. And so these all come together in our body every day, this sort of beautiful symphony of all these molecules working together to create all the activity in your cells. And so this is, essentially what we have traditionally thought of as things, DNA, RNA, and protein, but effectively this, is the central dogma. But we've historically not been able to sample it as deeply as we'd like. What has happened in the last really 10 years is nothing short of the fastest technological change in human history. So this is Moore's law, which is lets all of our phones in our pockets be faster than supercomputers 10 years ago. And now actually, the cost of sequencing human genome at one point was cutting in half every 5 months. So now you can sequence human genomes in almost any genome for much, much cheaper than you ever had been able to do historically. And so this lets us now interrogate any DNA or RNA of any cell of anything you like, including everything from subway to space, which I'll tell you about a little bit today. But this $1,000 genome is very exciting. People are talking about it a lot, but the thing I want to go back to is what we're doing with it is generally still along these lines. That you're thinking of DNA, RNA, protein. But the first part of the talk, I guess I want to say is, how do we expand this? I'm going to do essentially an average of 5 minutes per section and describe a variety of expansions to the central dogma. So the first is to think about not just DNA, RNA, protein, but of course epigenetics. And so it's not just enough to say that there is this progression of information, and actually we know there's a lot of regulation that happens on the DNA side. So specifically, how do you go from 4 atoms at just a methyl group, which you can draw a number of different ways, how do you go from this small group to actually a phenotype, and how does that manifest? So we've been looking a lot of this in really aggressive cancers and adult acute myeloid leukemia, and we can see that it can even define different subtypes of cancer. This has been defined since 2010. We've even seen other variations of this, like hydroxymethylcytosine, or variations of this base can also separate different aggressive states of leukemia. So there's this other layer of regulation that's behind the DNA, the methylation. And there's been a really interesting paper that's only come out in the last 2 years, and follow-up work has validated this, is that inside each one of us is actually an epigenetic clock. If you look at all of your cells, in each one of them, you can have there's a series of CPG sites, a place where methylated cytosine happens that slowly gain methylation and slowly lose your methylation. In each one of us, in all of our cells, is this clock that's slowly ticking away in each one of us, and then you can see it across different cells. And if you look in cancers, what's interesting is actually it's expanded, it goes even faster. But if you look in stem cells, you'll see here that it's actually, if you reset induced pluripotent stem cells, they go back to 0. So there's this epigenetic clock that's happening and ticking away in all of us, as we speak. It can actually, here's your predicted age versus your chronological age. You can actually predict with just a few markers. In theory, with just as few as 3 CPG sites, your exact age, which I'll come back to this later, that deals with questions of privacy. So this is normal cells or induced pluripotent stem cells or different cells in your body. If you look in cancer cells, the average methylation age is actually accelerated. So cancer cells you normally think of as being young, like they have telomerase activity, they can divide indefinitely, but in some aspects, they've shifted this way. They appear epigenetically much older than normal cells. And so this is a relatively new discovery in epigenetics that there is a sort of dysregulation of this epigenetic clock in cancers. And so we've been working on a lot of tools to look at this in the lab with this sort of expanded dogma of epigenetics. So particularly we have a tool called MethylKit and also one about different regional methylation. In the lab, the working name for MethylKit for a while, it's an open source R package, was called MethLab, but then we started thinking if we called it MethLab, we would have the FBI tracking us, which now the FBI does track me, but that's for different reasons I'll talk about. So we actually created this point to really think about how you can think about not just DNA, but the epigenetic data as a suite of tools that looks at these different sort of marks. And so I'm not going to talk about these too much today, but I want to talk about one that we just published that's very exciting, I think, as a concept before we start diving deeper into subways. And so meth clone is one that lets you look at epialleles and how they change. And so if you've not thought of this before, I want to just give you a simple schematic. And so going, think of DNA methylation happening across the genome. And if you look at three areas, they're both an average of 25% methylation here. You can see that they're an average the same, but clearly these are different states. So if you look at these are fragments of DNA, each one of them methylated or Y is unmethylated. They're clearly, on average, yes, 25%, but different sort of epipolymorphism or variation present. This is a great paper from Dan Landon in 2012. This got us thinking about if you take individual sequence fragments, when you sequence DNA, within this area, they're phased. If you have essentially, say, 4 CPGs together, you can actually get phase information, or what we call epialleles. And now we're going to look at the compositional entropy change between two states. Take two BAM files and compare them. And what you want to think about is that, an epiallele is really just one of a number of patterns. So if you take all of your reads and you stack them up onto a locus, like here, you can get essentially 4 CPGs in a row, sometimes within one read, even a 50 base pair read. And you can then count essentially what's the proportion of these epialleles per locus, which is a proxy for when you grind up all of your cells and take all your diploid cells, you can then count the number of cells that carry this epigenetic mark. So we started looking at this in some of the cancer samples to see, well, what are these epialleles? So essentially, if you have a yes or no methylation, and you have 4 CPG sites, it's 2 to the 4th power, or 16 possible states. And so we just counted them across the genome to see what we could find. And also we like colors, so we converted them to colors. We thought about clone wars, what's happening with clones. I'll come back to Yoda later. So in particular, we looked at one patient who was actually someone who cleaned the tanks at Chernobyl after the nuclear accident. So you can imagine this was not a pleasant job. And unfortunately, he got leukemia a few years later. So we want to look at this patient in terms of really deep sequencing, whole genome sequencing, and also looking at the clonality with the DNA methylation. But we can see in some cases, essentially this is from lymphoblastic cells, you can see before the treatment happened, but before cancer came, you can see there's a proportion of epialleles, a proportion of cells present, but then once the tumor came, essentially was taken over and dominated by this one clone that had taken over cancer. But in other areas of the genome, you can see selection happening in a different way. So in this case, you can see one epiallele, in this case the greener one, essentially expanded and took over the tumor. Then there was a bone marrow transplant, and eventually, unfortunately, the patient died after this last time point. But you can see that every time you look at a tumor, to some degree, at least epigenetically, you're looking at a slightly different epigenetic landscape. So we wanted to quantify this across the genome and say, well, what are the number of epialleles that you see that have changed as a function of the entire genome, what we call epialleles per million loci. And this is a Surcos plot, which everyone in informatics loves making Surcos plots. This just shows you each area of the chromosome in black across 6 different patients where we see different sites of significant clonality shift. And you can see some patients much more than others. Sometimes it's all black, sometimes much more infrequent to have epigenetic shift. But what's interesting in this paper we just published this fall is that if you take normal individuals and compare them at 3 different statistical cutoffs, the colors, the difference between one person within 6 months epigenetically can be different than complete strangers that are different ages. Even though epigenetics and age is a drift, you can see in some patients they don't change as much, but others are. What happened within 3 or 4, sometimes 6 months, they have a greater number of sites that are clonally selected in that patient than complete strangers. And so this means that this epigenetic state can actually change very fast. And we have a paper in review showing that patients that actually have a greater degree of shift actually relapse faster or worse prognosis. And so this is kind of an exciting new way to measure epigenetics that I want to quickly highlight. Now I would jump to RNA, because I did have single cells in the title, but I wanted to gear most of the talk towards what we've been seeing in the subway. But we have been looking also at single cell RNA sequencing. So in this case, we're using what's called a C1. It's a fluid MC1 system, where you can actually take cells, load them on a plate, and then look at each individual cell, make sure you only have one cell. You can C2 lyse them, make cDNA, and prep them, and then you can sequence them like we can sequence lots of things. And when we first got some of the data back to this, actually I was dumbfounded because we did a PCA plot, and this is what it looked like. They're very different in terms of their heterogeneity. If we just calculated high expression in red, low expression in blue, if you look at individual cells and compare individual genes on this axis, you can see that every individual cell is almost its complete distinct transcriptional landscape, which makes a lot of our work not necessarily, at least a little bit more difficult, because every cell has this complete heterogeneity that was more than we expected. But when we actually separated out patients and did this again and again for different patients, there is still a signature that persists within patients. So if you take, when you compare essentially one cell versus one cell, it's noisy. If you compare one cell versus many cells, you can see it's there to get a higher correlation. And if you even do different single cell experiments that were between replicates of experiments, it's not complete noise, but it is if you just do one cell versus one cell like you see here. But when we look between different patients, we can still separate them. So you can see this is what we've done now, over 1,000 different individual cells. And you can actually see we can separate out patients by individual patients. So even though there is noise, there is a sort of person-specific noise that we can tease out of the single-cell RNA-seq data. And we just got these in the lab, and we're doing this now before and after chemotherapy to understand what happens to the heterogeneity levels in these patients. So another area I want to just quickly mention is also the epitranscriptome, which is sort of a nascent word that we looked at a few years ago. In particular, is called something called the epitranscriptome, the sort of middle layer, which used to be a made-up word. No one believed us when we made this word, but now it actually is. It does appear in PubMed more than one time, so I'm excited about that. But what this is a large regulatory layer of RNA modifications. In the first paper we published on this, we could see that a lot of these areas of methylation right at the end of the coding sequence across all genes in both human and mouse. We see these peaks of methylation of RNA, not just DNA, but RNA being methylated, that we can see present across the different samples. And they're highly conserved, so we look at where the peaks are versus other areas within genes, in exons actually, and so we can see they're much higher, undergoing a lot of selective constraint. And so what we've now thinking about on these sets of RNA modifications is that there's ways to methylate RNA and then to process RNA to other various states like hydroxymethylformal carboxyl. In this case, actually, this becomes this cycle of turning RNA bases into different bases as needed in the cells, much like you do with methylcytosine. We talked about 5-methylcytosine and hydroxymethyl. This was on the cover of Cell about a year and a half ago. It's the same concept that DNA and RNA are both undergoing these cycles of base modification and regulation that you can see here in the same concept. So what we looked at is methyl-6-adenosine, which is one out of 110 modifications of known RNA modified bases. So the amount of work left to be done, this will be a recurring theme for this talk this morning, is that we've looked at one modification out of over 100. And the same thing we'll talk about later is that we've started to discover a lot of organisms, and we really have just scratched the surface. So now when you look at the central dogma, I think we add lots of things. If you want prefix consistency, I would say all post-translational modifications you could call the epiproteome, but that only appears in PubMed twice. And so that's still a relatively new word. But in sort of the concept that information flows not just forward, but also goes backwards, that there's inheritability in some cases of epigenetic states. There's evidence that viral-induced microRNAs are also heritable. And then even prions can go even between species, and prions just kind of freak me out, a little bit creepy, I think, and they can just, they're a little bit disturbing, I think. But in the context of other information that can actually transmit between generations or between species, it's not as creepy, I guess, in that context. So that's a bit of a background on sort of central dogma, which I wanted to just discuss about how it's expanded. But now I want to get into what else we're doing with having people come and help with, let's say, citizen science. So with this, this area of personalized medicine, essentially people sequencing genomes for cheap, people have started to even just share all of their information online. Jamie Haywood's site called Patients Like Me, you can go in and browse. People put all their medical information online, essentially what's their mood, what drugs they're taking, what's the efficacy, you can go browse them. People can just band together. So for example, if you have, say, Crohn's disease, you can look at everyone who has Crohn's disease, what drugs they're taking, see how they're doing. Using sort of this big data approach to personalized medicine, you can see what's the rating of side effects, what's the ranked effectiveness across all the patients who are logging on. Even at Cornell, actually, when you get blood work done, this is what you can have come back to you every time you log into your own personal account. So my favorite thing about thinking about this world of sort of personalized quantified medicine is actually emails from patients, is that patients really feel enabled and empowered. They also feel like they get to monitor their body in ways that they've never had before. It's not always clear, necessarily what it means to the patients, but in a lot of cases, they are really excited to be able to look at the data. And for example, in this patient with a high percentage of different cell types, he could actually see that was causing him to have itchy skin. So in this case, the brain is reinterpreting its own perception of its body's phenotype by having more information, which in this case is soothing and was helping this patient who was a person who was emailing about his genome. And so in this case, you could make the argument that sharing is caring in this case. For example, I've shared my genome online, and I'm even more sure that I should not smoke. So I don't, and I'm at high risk for lung cancer, so I don't smoke. And you could even say that at Cornell is even this idea of having genome-guided medicine, which we're doing now for every cancer patient. So people are even sharing all of their data. A lot of people are just sharing essentially part of the global alliance for genomics health. Every genome and every medical record that you can is being shared. The idea that more data is more power is what's the goal effectively. So this also blends into citizen science, sort of participatory medicine, or people who want to actually take, for example, their own DNA or their own resources. So in Brooklyn, there's actually a lab where you can buy a lab membership, much like you buy a gym membership. You can go there and do experiments. And so here, for example, if you want to go sequence some of your own DNA to see if you have mutations in LRP5, you can go do so because you could have really, really strong bones, much like Bruce Willis in Unbreakable, if you want to see if you have sort of like superpowers. You can also go there and take one of their courses, which is to look for mutations in CCR5, which can give you native immunity to HIV, and that actually is what they do for one of their courses. So I actually brought some friends of mine to this course and said, I want you to come to GenSpace. I'm going to take you there. It's your birthday. So it's your birthday. For your birthday present, I'm going to buy you this gift certificate for a one-day personal genotyping workshop, and come on down there because I'm a big nerd. That's why I did this. So what's interesting is you can see most of their reactions didn't work. You can see here they tried doing PCR and pipetting for the first time. It didn't work for most of them, but for the ones that did, we could actually, none of them had the mutation that would have given them immunity. But as we were doing the experiment, I said to them, actually, this would be infringing patents as we're doing this in 2012. I said, well, what do you mean? I said, well, if you look at, who owns your DNA, as soon as you take it out of your body, at that time, it was considered actually be infringing those patents. It's, you would be violating essentially their property as soon as you take it out of your body. And that's because essentially thousands of genes have been patented essentially since the late 90s and even the late 80s across the genome. And depending on what threshold you use, anywhere from 40% to 100% of genes all across the genome have been patented since the late 80s. So we challenged some of these actually in court. I worked with the ACLU to actually challenge these patents on what's called the myriad BRCA patents. And particularly, it's these two claims, not only the gene, but also the cDNA. And if any of you have done an alignment to a genome, which I think everyone in this room probably has, they also claim things like this. Not only the gene, but any 15 letters of that gene that matches any other gene. So you can imagine how non-unique this is. And in fact, that's what we showed, is that there's no such thing as a gene that doesn't have at least one 15-mer that matches every other gene in the CCDS database. And so it's not possible to only patent 15 letters of a gene. You actually are patenting lots of genes this way. And so this is one of the bases for the challenge of the patents and what we put through court. So it went through district court, went all the way up to the Supreme Court, and essentially after the oral arguments in 2013, they were challenged. I saw Eric Lander and Francis Collins high-five each other outside the courthouse. They were very excited. And then, as most of you probably know, that actually a lot of their claims were rejected, and now a lot of litigation is ending just this year. And so I would say that this is kind of, in my own mind, prompted this idea of what are sort of rights. What do you have that's your genomic rights, which can include their rights to, say, share your genome, but also other things. So I think you should have the right to look at your DNA if you want to, or to have your doctor look at your DNA, or the right to even modify your DNA. Let's say, for example, that you really like, having a shower in a green lit room. Let's say you want to take your cheek cells and make them, say, GFP positive and have them glow next to you in the shower because you want your shower to be green. I think that's your right to do so. It's your DNA. I think it's your right. If you want to PCR your genes in a different context, or if you just want to interpret your genome and all your owns, or if you want to just convert your genome sequence to poetry, which I also did here with this haiku. So I think this, which technically infringes one of Mary's patents. So I think this is your right to do so if you would like to do so. So your genome is now free. So this is kind of exciting, or at least some of the genes in it are now free. So it's a very exciting time in genetics. But there's more than one genome. So as most of you know, there is the microbiome. That's the human genome. It's exciting. It's a bit more free. But in the cellular democracy that is your body, you are actually a minority party. So if there was a vote to see where you should go and walk around, you would always lose the election. You would never get to decide. And so you just generally wander towards probably tepid pools on the ground or something. So it's because you're number 10 to 1 by bacterial to human cells. If you look at active transcripts, it's a much bigger even ratio of number of active molecules. And you can separate patients with Crohn's disease, ulcers, or healthy patients by the sort of microbiome. So there's been a lot of excitement about microbiome research. And again, here's this schematic. This is human cells. Prokaryotic cells are much smaller, but human cells are big, and we're covered in bacteria and many types of bacteria. The big question is, what are they, and how much are there? So actually, if you add up everything, if you step on a scale, about 3 to 5 pounds of your body might be bacteria. So it's a lot. It's like an organ essentially in you. So if your diet's not working, it might not be your fault. You could blame them actually. And this has spurred a lot of research on the human microbiome project that's looked at what are different areas of the body, stool, cheek, tongue, nose, vagina, skin, that are colonized by different normal flora, just normal microbiomes that are present in and on all of us, and what is sort of the diversity of the microbiome and what is it doing. So this has prompted a lot of excitement because not only what they're doing there, but what they're making. A lot of the serotonin in your body and dopamine actually lives in your gut. You could argue this is the 2 brains in 1 body problem or solution. The microbiome is linked to cancer in terms of creating a persistent state of inflammation in some cases. It's also, essentially led, news reports to just start saying, well, you just are your bacteria. So forget about the human cells. You just are bacteria. This guy will forever be associated with obesity, this poor guy. And even Lee Hood has said, if you think of what's in your bloodstream, he's estimated 1/3 of the molecules are actually, from the microbiome. So if you're looking for the nearest pharmacy, it's not down the street, it's actually in your gut. So this has led even to look at examination of what's present not only in your gut, but in other areas of the body, from Andy Fishbach, Michael Fishbach's lab and Mohammed Dania, of saying, well, actually, it looks like we can see biosynthetic gene clusters, so what's being made by these microbiomes. It looks like in some cases, there's entirely novel antibiotics that are being made by normal flora that resemble, in some cases, a lot of what's being developed in pharma, that these are sort of new biotics that were discovered, but that have been in us all the time. And so there's many drugs inside of you right now that are just being made by normal flora. So like this, a lot of people think of microbiome hope, like this is amazing. There's natural drugs inside of us. This gives people a lot of hope. This led to the large-scale projects of the IHMC and the HMP, and said, well, you can actually treat obesity this way. You can do a transplant and get skinny mice to become fat, and the inverse, essentially, when you use transplants. We can start to understand how fast the gut changes. We can start to think about other sort of inflammatory bowel disease or other potential diseases, which includes this list right now of everything that might be to some degree influenced by the microbiome, with the molecules that they make or the impact on your cells. And so this, the big question is, well, where does it all come from? And a lot of it comes actually even before you're born. This is actually, in many species, there's something called maternal microbial transmission. So the placenta has its own rich microbiome. Previously thought to be a sterile organ from the trophectoderm, but actually it has its own microbiome and is contributing towards fetal development early on. And there's actually, of course, a vaginal microbiome. You get a lot of antibodies as well as microbiome from breastfeeding breast milk. This has even led Rob Lang, when he's doing his TED Talk, about the risk if you have a C-section versus vaginal birth is higher for a variety of diseases later. So actually, after his child was born, he actually took the vaginal microbiome of his wife and then coded on the baby in the OR, which actually was the first time it was done at that hospital. But now there's an ongoing trial at NYU in Manhattan to try this, and mainly because he wanted to create this idea of the microbiome, will really give sort of that force field right at birth to protect essentially his child going into life. So, by the way, actually, Boeing has recently patented force fields, just as a quick sidebar. I can't even make this up. They really did. This is their patent. They patented a force field just last month. And so I don't know if it even really works that well, but if you want to check it out, come on. But in this case, sharing here is protecting. The idea is that you want to share the microbiome because it's protected. So the other thing to think about is what else might protect you, not just the human microbiomes, but other sort of exposure to antigens is generally a good thing. So if you look at the odds ratio of having sort of wheezing or asthma later in life, if you get more exposure to these things, which normally you try to avoid, cockroaches, mice, but cats and dogs, people like, it's decreasing your odds ratio of having asthma or wheezing later in life. So you need the exposure, and especially in early development before age 1, to have sort of this exposure to the environment, or else you actually have a higher risk of some of these diseases. So in this case, sharing also is caring, even from other organisms. So the sort of ultimate, though, of sharing and caring, I'd say, is something called fecal microbiome transplants, which if you haven't heard of these, they are exactly what they sound like. So it's, and a lot of times for treating Clostridium difficile infections, there's been a lot of hope, a lot of excitement, because they've been extraordinarily powerful in actually treating infections. So people saw a lot of the work that was happening in the literature, people treating themselves, and essentially they started doing it themselves. So the Scientific American had this sort of unfortunately titled article about fecal transplants, saying that, this is, wait a minute, people are doing it themselves, we need to regulate this, we need to do trials. And essentially this is because websites like thepowerofpoop.com, which I can't make this up, this is a real website, that they were saying they want safe, accessible fecal microbiome transplant for all who need it. So you can go to the website, you can learn about FMT, and you can learn about, well, literally the power of poop. But it's amazing because thousands of people have been treated, and successfully. So that's why patients, participatory medicine, people just doing it themselves, have led also to the creation of something called the open biome. So in this case, people sharing their biomes with each other has been happening. And so really, what they've argued on their website is it might even be the most important thing that you do all day, depending on what your size and how many people you can treat. So they even had a campaign to get people to donate. And they raised a bunch of money. So I emailed actually Eric All, who ran this campaign. He's in Boston. I said, I was updating my slides and I found your website. It's amazing. He said, well, sharing is caring. He was like, this is really, really good. And then I also found this article about his work, and I said, Eric, how many emails did you get after this article came out? And he just said, you have no idea how many people emailed me. But what they're doing is quite serious, actually. They're taking it, they're checking it for any known pathogens or diseases, but then attempting to do C. diff treatments and doing fecal microbiome transplants. And so far, it's over 4,000 patients. The vast majority of them have been successful in terms of their transplants. So it's really exciting sort of time of research, and something that maybe 10 years ago would have been completely laughed at, and now it's just a little bit giggled at, but at least it's actually a very serious health impact. But there's other questions. So is a fecal transplant a drug, or is it a tissue? What are you giving, and how do you regulate that? Depends on various questions, which he's looking into as Eric Holmes' work. And generally, if you want it to be a tissue, it's much better for regulation purposes, which for the most part it is. So this leads to, that's the hope, but then of course there's some people, you think of microbiome, you think of bacteria, a lot of people are not as excited, they'll think of microbiome fear. So in this case, if you think around the world and you're worried about, say, outbreaks of different diseases, you'll say, well, what if things start emerging in Africa, or started seeing things in the Caribbean? There's everything that happened with Ebola recently, of course, most of the news. But this is what people think of usually when they think of microbiome or outbreaks. They're terrified of bacteria, they're terrified of viruses, even though the vast majority of them are fine. And even if you go on the subway, you'll see signs that look like this and say germs are everywhere, you have to watch out, which is true. You should wash your hands more and sneeze into your arm. There are sort of basic public health features that are unquestionably true. But there's other people that are just seeing papers that look at what is a bathroom? Everyone's scared of surfaces in bathrooms. There's even a study looking at what does a bathroom look like on the microbiome level? And yes, men's and women's bathrooms are really different. You can see here so your suspicions are always have been true. A lot more lactobacillus in female bathrooms, which is likely the natural flora of females. And but what's kind of terrifying about this paper is that over time, really, even after you clean it, eventually just the bathroom, sort of on the surfaces, just end up looking like the floor. And so really, which just has a lot of more fecal and skin-related bacteria. Because remember, a lot of the toilet seats don't have lids. So what you have is just an aerosolized, sort of automated aerosolizer in every bathroom around the world, especially places that don't have lids. So that's not surprising if you think about it, but people generally just get terrified of it. And you go to, say, a hotel, you might see remotes like this that are easier to clean because they're hermetic, they're sealed so that you can just actually quickly clean them, and everyone's afraid of their remotes even. I know some people that when they go on traveling, they don't sleep on the bed. They bring their own essentially backpack and their own blankets because they're terrified of beds and bed bugs. If you go to maybe you're at the hotel and you go down to the pool, you might see signs that look like this, which is true. But why is that sign there? I didn't want to go swimming in that pool, but this is again part of sort of the fear that's out there. In this case, sharing's not caring if it's in that pool. But you go to the airport after you leave the hotel, you might see signs about H7N9, or you might see signs about MERS-CoV, all these signs about different viruses that are in the environment. You go on the subway, and you'll see people do this, like this really, in this case, a very nasty-looking paper towel by this woman who didn't want to touch the surface. I even caught one guy with a glove, like a lab glove. Like, did he leave lab and just keep the gloves on? I don't know, but I saw this in the subway. And so a lot of this still goes back to, why I'm curious, though, I don't want to leave the house in a BL4 suit. I want to just know what's there. And so I'd rather have, instead of being afraid, and if people are looking around the environment feeling like they need to wear gloves, or you're afraid of everyone who sneezes, wondering what's in the air, I'd say that the danger here is that, fear, which people oftentimes have, can lead to superstition. So then you won't touch certain things, you won't look at people certain ways, you start to become paranoid. It might make you to be mean to people, and so essentially it's not a good world we're creating. But as Bertrand Russell said, to conquer fear is really the beginning of wisdom. So I like to, whenever possible, replace the fear of the microbiome with actually knowledge of what's there. And more particularly, when my daughter actually started riding the subway on the way to school, this sort of seed had been planted in my mind that, what is actually there? And there was no knowledge. No one knew. I asked people looking in the literature, no one actually knew. And even before she started riding the subway with me on the way to work and dropping her off at school, is actually the first time I dropped her off at daycare. You sit her down, exploring with her mouth. She's 6 months old, and she puts a toy in her mouth and puts it down. The next kid comes by, and he does the same thing, puts a toy in his mouth, he sits it down. The next kid does the same thing. And all the kids are basically making out through this toy proxy in the daycare. And I just thought, in the HMP papers that just come out, I thought, what if I swabbed her mouth when I drop her off? And then at the end of the day, I can swab her mouth again and see what microbiome has changed. I'm sure it has changed. I just wanted to know what it was. But then I would be that creepy dad running around with the swabs. And so I couldn't do that. But this seed had been planted, and I thought about it for a few years. And so finally, actually in summer of 2013, we just started we got a sort of a gang of grad students and undergrads. And we said, let's just start actually swabbing everywhere. Let's just start seeing what we can find and go around. So actually, and I would even argue that maybe one of your rights could be that you have the right to sequence the DNA of any surface that you touch. I'm going to add this to the list and say that's one of your rights, I'd say. So to that, we started to create sort of the city-scale metagenomics, or to look for molecular echoes and what we could find. A project was launched called PathoMap. And the goal was to look for pathogens and to see what's on different surfaces. But we also wanted to understand the urban biome of what's present. And so really, we want to create, you could call maybe metropolomes, another word for it, so New York called it this. But we really want to create this sort of molecular view of the city and to really understand both good and bad sort of microbial features and to track and classify, identify them. But really, you could argue we want to actually seek out new life and new civilizations, I would say, what's around us. So what we built first was an app, open source app. You can load in the data as you're collecting it. So it populates on our database, and you can visualize it at pathomap.gs5.com. You can see the data as soon as it's being made. It's actually great when I'm traveling. Actually, today, if you go to the website, you'll see they're swabbing the Gowanus Canal, which I'll talk about. So as we're here, you'll start to see data points populate as they collect more. So we can see what have you swabbed, how long do you swab, take a photo. There's actually data redundancy. So you have to take a picture of the swab that has the same label ID, and we make sure that we only process samples that match. And so essentially, we also made a volunteer kit. So we had a few volunteers help us. We made this little packet. And we also had the granola bars in case you get hungry. So we put that in there. And so people are volunteer packets. We only got a handful of volunteers. We were hanging around on the street. People are generally confused. But we did get some volunteers. Mostly it was student driven, though, and volunteers. So this is how it started in summer 2013, a few data points, and we basically have been going ever since. We finished now 6 different seasons, essentially sampling and sampling. We're just starting now spring of this year, and at this point now we have over 4,000 different data points across New York City, and in some cases places around the world. And yes, I will be swabbing the Baton Rouge airport before I leave here today. So what we did is to get all the first set of data, and actually we just published it 2 months ago, which is sort of the first city-scale metagenome profile, which was on the cover of Cell Systems, and was sort of the first map of the city. And I was excited, it was on the cover of the Wall Street Journal. What is in the city? So what do we find? So you're curious, okay, it's fine, Chris, but what's actually there? So the first data set is over 1,400 samples that represents what happens when 5.5 million people ride a subway every day over 656 miles in 468 subway stations, also up in triplicate. So what this is the data entry method I just told you, but then also we took them all, extracted DNA using the mobile kit, did TrueSeq and QIAGEN libraries. It's over 10.3 billion Paradigm 125 DNA fragments on a HiSeq, trimmed them, did Megablast, and then also did a confirmation with Metaplan to see what was there. And so what we found, my favorite result of the study is that literally half of the DNA that's under our fingertips matched to not a single known organism, using fairly loose blast criteria, 80% match over sort of the 85% query space. So that's, it just doesn't, you can bring the thresholds lower and you can match to anything, but this was actually fairly loose already, and we couldn't make a match. So we made all these maps of what we could find, which mostly was bacteria. But you can see a little bit of eukaryotic, a little bit of viruses and archaea, but most of it was bacteria that we could match, but a lot of it was really just unknown. It's all around us, which we're probably new to this room as well. So of the things we did find, if you Google now microscopic pizza, this will be the first hit, is that some reporters said that the subways are covered in microscopic pizza, or Enterococcus italicus, which is not necessarily wrong, it's just not really true either. So I like this headline. And the other thing I like is New Yorkers are a little bit acerbic, they're a little bit kind of mean sometimes. So like this guy's like, is that dude wearing a Starfleet uniform? And it's just, that's Captain Sisko's far less successful cousin. These people are mean online. But then some people thought that 48% was aliens because we didn't match their non-organism as aliens. But really it's just the incompleteness of the phylogenetic trees. And what we actually saw is the majority of them are harmless, that they are have never been associated with a human disease or are very unlikely to ever get used to it unless you have a reduced immune system function. But there are some things that could be associated with disease, like Shigella we found, or some Enterococcus faecalis, which is actually a fecal-related bacteria. But some are only fatal to lobsters, like here, like Enterococcus viridans, but a lot of them are actinobacter or lactobacillus, or things that you just normally find on skin. And so most are harmless, but some things we did find. So in 2 samples, the top hit with every tool we used was Bacillus or anthrax, and on a few other samples we found hits to Yersinia pestis with the plague. So this led to some interesting headlines about what was actually there, and people really freaking out about what was present. And what's interesting is with every tool we tried, Metaplan, we tried Serpi, we tried Blast, we tried BD-William with specific alignments, they all just predict and say it's a high confidence hit of anthrax. And so we said, well, here's what the tool said, and here's what the coverage plots look like. And so, but as I did a follow-up, because there's a lot of debate online and on Twitter about sort of what did we really find. And so, just because you have DNA doesn't mean it's even alive, which we said in the paper. And even if it's alive, it doesn't mean it's pathogenic. And even if it is pathogenic, doesn't mean you can still catch it, because it depends on infective dose and other factors. And so we just claimed with what DNA and the closest match that we could find. But we tried. This is an ongoing debate to some degree in the field about the incompleteness of the phylogenetic tree. And when you have a best match, the debate has been, should we have still reported it or not, given sort of public health concerns in New York City. My argument's still been that I thought it was irresponsible to not report it. These are standard methods, standard tools that everyone in the HMP uses, everyone in every other microbiome paper uses the same tools. So I think it's a good debate to have about what are they finding when you take environmental samples. What will you find? Bacillus anthracis after bacteria and the whole Bacillus sort of family or genus. So in any case, that's a longer debate which we could have over sort of beers and coffee later. So, but things that we could find, other questions we looked at was diversity. The Bronx actually is the most diverse in terms of average number of species found per swath station. I'm coming from Brooklyn, so I was hoping Brooklyn would be number 1, we did not. But this kind of makes some sense, because nothing soft comes out of the Bronx, as we know. And also Brooklyn's beloved, that's why we're number 2, I think is what makes some sense there. But as I alluded to before, we looked at what do they match? If bacteria we could match, what's the associated bioregion from the HMP data set? And we can see this is what things match. But then you have to account for what's in their database versus what the things we could have found, what we observed. So it's a log ratio of observed versus expected. We can see it's almost highly enriched for skin and airway-related bacteria, as well as what the subway looks like to some degree. And then also, Wall Street Journal made this great map where you can zoom in and out. If you want to see what the bacteria associated with kimchi or sauerkraut, you can zoom in and out and look all across New York City where they are. If you're so interested, or if you want to see where all the microscopic pizzas are, you can play with this map. And we also made some of our own maps around on pathnmap.org. So The other thing that's interesting is, there was different diversity across different areas of the city, but one thing that's really interesting is some areas had very, very unique bacterial signatures. In particular, one was South Ferry Station, which was the one that was flooded by Hurricane Sandy in 2012. And so what's interesting is there's 10 species here that were not present in any other part of the data set, and we actually have not seen them again since. They're unique to this station, and what they are is actually sort of marine-related bacteria, which represent sort of a cold molecular echo of this ocean water that's still present. In particular, some of the species are like Shirmonella marina, which is actually previously thought to be only an Antarctic species, found in sort of cold water areas. It actually makes this kind of acid called EPA, which you usually only find with fish or with salmon. So even the fish don't normally make it from what they consume. And interestingly, if you don't have enough of this in your diet, you're at a higher risk for suicide. So I made the comment once that in theory, some of the bacteria could be beneficial. Like you could even lick the walls of this subway station, and it would potentially be beneficial, which then led to a number of comments by people on the media. And then I mentioned that I wanted to stand by the point, though, because in most cases, exposure to antigens, as I talked about earlier, is actually a good thing. So I said, really, again, this is the danger of making an offhand comment when someone who's from the media is anywhere within earshot, is that really, depending with newborns, what you really want to do is really get them to expose to different antigens, which I still stand by because NEJM just came up with a paper that showed, like 2 weeks after our paper came out, is that actually the worst thing you can do for someone that shows a peanut allergy is to take away all peanuts, actually. If you keep exposure, low-level exposure to antigens, you'll actually reduce the chances of having a peanut allergy later in life. And so I sort of stand by this. I still think, I'm hoping they would go with the spirit of my words, not the exact quote, but in any case, the concept is the same, that you need the exposure. You can't, the hygiene hypothesis rests on the fact that you need this exposure. So the other things that we look at in the data set is, well, how does it change over time? What are the dynamics? And so we can see that, we look at one kiosk where you buy your tickets, depending on what time of day we look at. This from Penn Station. So really, Penn Station, we got a lot of DNA, and Grand Central much less DNA. I think they just cleaned Grand Central a lot. If you've ever been to Penn Station, you would not be surprised. It's like sort of an armpit of the city, I'd say. But in this case, we saw rapid bacterial diversity shifts throughout the day. Pseudomonas was present at all time points. And we actually even made a little video where you can see, and it happens at what time of day that you look, you'll get different proportional bursts of different kinds of bacteria. So depending when you're going to buy your ticket, you'll get sort of a different microbial profile at those different stages. You can see the time at the top there. So it means that there's this rapid dynamic of really tens of thousands of hands touching these surfaces. And so what you'll have is the equivalent of shaking hands with 10,000 people a day. Some politicians do. My brother's a politician. He likes shaking hands with 10,000 people a day. If you don't like people, that's a terrifying thought. If you think, I don't want to touch 10,000 people, you might be afraid of that thought. But if you think it's sort of the brother and sisterhood of humanity, it might be a soothing thought. It just depends, I think, on if you like people. So you might wonder what's in the Gowanus canals. I mentioned we're doing more sampling in there today. So this is the canal in New York City that was so heavily polluted. The water, yes, looks like this. It's just kind of an eerie color. There's a few fish that have managed to survive in this water. But what we saw here actually is interesting is a whole subset of archaea that are actually processing the sort of methanogens that can process a lot of sort of methane and create a lot of methane and use sulfur-reducing compounds as part of their biochemistry. And so we can see this unique sort of archaea signature that's present in that canal that we also didn't see elsewhere in the city. So you can tease that out from the data as well. So essentially it's a sort of a methanogen heaven, I'd say. The other thing that we looked at in the data was, do we see any evidence of live or antibiotic resistant bacteria? So it's just DNA, but is it actually alive? And so actually the answer is yes, that in a lot of cases, about 20% of them, when we tested, we did see antibiotic resistance. But then took these plates and compared them and say, if we culture them here on 2 different media on the right, or just take them and sequence them, what's the overlap of species that we detected? You can see we would find more species by sequencing rather than culturing, because the culture process enriches for only things that will grow on there. But the thing I really like about this paper, this is from figure 4 in the paper, is that we treated them with tetracycline and said, well, can we do before and after treatment, can we pull out what might be driving resistance to tetracycline? So here, we did coverage of known tetracycline resistant genes before and after, just a ratio of what is sort of the coverage of different genes. And TETK, in this case, on both media, is apparently what's driving the resistance to tetracycline. So that's what we showed here. There's evidence of using metagenomic tools to pull out the likely genetic drivers, at least for tetracycline. So this is ongoing work that we're starting to do in the hospital. So you might be wondering, well, okay, that's bacteria. What else is out there? What are those eukaryotes? What's really there? What was the other DNA that was left behind? So in this case, we saw potential hits to a lot of things, including all these potential organisms. So we really, humans were number 3 on the list of what we could find, but we also saw, cucumber and chickpeas, which I just think might be falafels in the subway. So we saw low levels of all things that would match various plants, but its blast results in that low confidence. And so the biggest challenge in the field, actually, I'd say, is not only the fact that the databases are incomplete, or different between the ones in Europe or the ones in the US, but that, when you do BLAST, you'll kind of find the closest hit, which just depends on which genome was sequenced that someone was interested in. It's not necessarily what it even is, and we say this in the paper, it's just what you can find as the closest match. So we do see things like chicken, maybe someone's having a chicken sandwich and they just touched the subway afterward. We saw a rat and mouse DNA that was present and zoom in on specific regions to see good matches, so it's down there. So sometimes when you're in the subway and you get that puff of air and you close your eyes and you're excited about being in New York, it's just rat dander that's coating you like snow. But you're all fine, you're all healthy. But then there's the human sample. So I had this hypothesis going in the data set of like, okay, so human DNA, we should get some human DNA, and some days in the subway have more human DNA than others. Actually, one day a year, there's a no pants subway ride day. It's by Improv Everywhere, it's a sort of comedy group, and suddenly the best thing is tourists who don't know that it's no pants subway day are taken by surprise. So there are people who are underwear, it's not no pants, nothing at all, but it's just it's a bit more human skin you'd expect on those days, for example. But from John Weber and Carlos Bustamante, they've shown that you can actually predict your birthplace based just on genotype data. You can actually remake the map of Europe just from SNP data alone. And so one of our questions was, can we actually do this same idea with the DNA left behind? You can even use these kinds of methods to predict it just from gene expression data. So you don't need SNPs. Since EQTL, since genes are controlled by some of these SNPs nearby them, you can even use the expression of genes to predict your ancestry. And also that your gut microbiome is also influenced by your ancestry. There's been other work that's been done in public service, like Heather Dewey Hagberg. She actually took DNA left on cigarette butts and then predicted hair color, eye color, skin color, and then made these masks of what was present sort of on public service in New York City. It's showing power of ancestry informative markers. And also, of course, the FBI uses these loci for their own CODIS database, which you can use to even track crimes, even if it's, even if you commit a crime but your brother's in the database. In this case, this is a guy who was caught because his brother had committed a crime. So if you have any members of your family who committed crimes, you should not commit crimes. You shouldn't commit crimes anyway, but you'll be likely to catch on what's called a familial DNA search. So you can do a lot with DNA in terms of searching and matching. So we just pretended that these samples, environmental samples, let's just pretend they're human samples, run them through our normal human variant calling pipelines, call variants, and then intersect them with known ancestry informative markers. And then go back and look at the map of New York City, which actually you can go to this, there's a great site on Cooper Center. Oh, by the way, I've tweeted my slides, so they're all available online. I meant to say at the beginning, so if you're worried you're going to miss something, all the links are online. And you can see white, black, Asian, Hispanic, different areas of the city. This, for example, is Chinatown. You can see this area near Canarsie. You can see sort of this is Harlem. You can see different areas of the city are sort of represented by different demographics from just census data. So we want to intersect the census data and see how does that look relative to sort of these regional differences of some machine learning algorithms to separate out regions, and then start to look. So if this area looks like a little bit more like a white area based on census data, what does it look like if you just predict the ancestry? So we actually see here the top sort of ranking predicted ancestries where British, Finnish, and Tuscan seem to be really matching sort of the expected ancestry. If you look at this area that's more Hispanic near Chinatown, we saw the same thing, predicted Spanish, Colombian, Mexican, with an increase in sort of Asian alleles at that area. If you look at other areas, you can see that there's Chinatown. This is essentially just north of Harlem. You can see it's more black and Hispanic. We can see the top ranking alleles at that station more African American and more African alleles based on sort of 1,000 genomes data. And so in general, I was actually shocked. This root mean square and standard deviation of what we saw in some cases was almost a perfect prediction based on the census data. So we were accurately measuring who lives in that area. In other cases, that was much more noisy, or maybe someone came here who didn't live there and was much different from people who actually carried those alleles normally. But overall, it's the whole data set from figure 2 that just shows that you can actually detect a human molecular echo of what's left on DNA in the surface. So in theory, we could take all the DNA from this auditorium and predict the likely hair color, eye color, skin color of everyone in this room, which is obvious implications for privacy. So in particular, I'll share this video. This is from Jessica Green. This idea of sort of what's left behind when you think of sort of a microbiome world, and you kind of once you've done this, you can't see the world the same way. That you know that essentially DNA is being left behind everywhere you go. This essentially you can see coats of microbiome, essentially fragments of bacteria present every time you breathe out or breathe in. Of course, you may move DNA around or different cells of dust that's left behind. So this is a video that she made to kind of think about what if you look at the world sort of as a microbiome. Your skin is, of course, coated in its own microbiome. You can see it's present in you, on you. You can see here's your GI tract. And so if you're working in an office, you're not just kind of hanging out and living there. You're actually leaving, to some degree, a microbiome aura that's behind you that you can detect not only your DNA, but also your bacterial DNA. So this is what Ed Young and Jack Gilbert call microbiome aura, or what I guess we call molecular echo, or some way to determine there's a trace left behind of what happens when you move through a room. So there's a lot of ongoing work and questions now about how do we design buildings or think about buildings. And since this is happening, there is an ecosystem. And if that's the case, how do we actually design for the ecosystem since we know it's actually there? So this is an ongoing question kind of in my lab and in the field, I'd say, in general. So it also gets the question of what if you don't want to leave your DNA behind? There's actually a website where you can choose what DNA to leave behind. You can order these parts where you get a little preservative and you can get a little spray bottle and leave sort of fake oligos behind, which you can be invisible. It's from how to do hack work again. So I would add that to the rights. Let's say you want to choose what DNA you leave behind. I think that's your right to do so. It might be, if you're committing crimes, this might get you back in the crime business if you want to commit crimes again. It's going to help you in theory. But so in the last sort of 10 minutes, I'm thinking about sort of what's next. That's what we've done. The first question people say is, well, okay, should I ride the subway? So the response actually online has been some of my favorite things to read. Is like people saying it sounds like it's the ultimate destination for building natural immunity, which I 100% agree with. Is what I was hoping for with sort of the spirit of my sushi comment, which is what you want. But then other people, really have a more conservative view of what they need to do for protection on the subway. They're a little bit more paranoid still, which I'm trying to have not happen. Other people think I maybe work for Uber, which I do not work for Uber. I take no money. It's not, I don't relate. So, what I want to think going forward though is, yes, you should ride the subway. Ideally, just with ice cream is really the answer. The vast majority of things are not harmful and likely not to make anyone sick. And what's more important is it's not as if it just sits there. They do, I can tell you, there it is. So they do clean the subway, not every system every night, but they do clean most of the subway system on given nights. And you can see here they scrub down the surfaces, the floors. You may have even wondered sometimes, can I lean against the walls of the subway? They actually do scrub, the walls, the signs, and so it does get clean. It's not as if it's just sitting there in sort of a festering pool. This guy loves cleaning signs, and so it does get clean. What this means actually is every day what happens is they come through and they'll clean the subway this way. What this represents to a large degree is really a forest fire of what was present for the microbiome. So they clean it, they wipe it all clean, and the next morning we all descend back down to the subway and we repopulate it with our own sort of microbiome, and things start to be redeposited and grow back again. So in this case, we know that this is happening, and so the question is, can we track it better? Can we understand it? Could we even design potentially a world that takes this into account, or at least monitor what's changing? So this is true, not only for say New York City, but also this is also true for Washington D.C. So I just keep track of things anywhere I travel and see where do people clean. So I don't even know actually if this guy works for the subway system. He doesn't have any vest. I just caught this guy mopping the D.C. subway, and I had to take a video. But I think it does get clean. I think mopping is probably just a way to move things around from what we've seen in our sampling. So I don't recommend it as a cleaning method. But to this point, we're just launching now is to build kind of this global context. So New York City is only number 7 on the list of busiest subways in the world. There's a lot of people in Asia, so there's a lot of busy subways in Asia. So we're just launching this summer. There's going to be 2 meetings in New York to try to plan this idea, but across many cities around the world. Something we're calling MetaSub, or Metagenomics and Design of the world's subways and urban biomes. And so this is something that we're just launching this summer. That's mainly as a real exploratory effort. It's been just funded by the Sloan Foundation to understand what's there, because again, there's almost zero knowledge of what's actually present. So we're going to start to build this map over the next few years. And so swabbing has begun in some areas where you see in green. Others are planned or pending, depending on where they are. So if you like swabbing, let me know. So this is a discovery approach, as we want to know what's there, but there's also the ability to bring it back to what we would say, I've worked at a medical college after all, is what we're doing actually in the hospitals to make what we call precision metagenomics. Think of precision genetics and precision medicine. But why is it that we take infectious disease patients and we wait 5 to 7 days to culture something, or sit around when things may or not be plated or picked off the plates and cultured in pure cultures, when we could actually sequence the DNA and know what's there, especially as it changes, especially to find antibiotic resistant genes like we showed here in this paper. We want to do this essentially for every infectious disease patient or a patient of an infection of unknown etiology. It could be fungal, it could be bacterial, it could be viral. You take all the DNA and RNA out of each profile, because we can now do it very routinely. And so this is part of also the hospital microbiome project. This is from Jack Gilbert's work. So we're doing it at HSS and at Weill Cornell, and track with present in the hospitals as well. So think about how do we change what we're doing in our hospital setting. And so the idea that we should have is be able to take a sample of any source, try a variety of different methods and a variety of different sequencers, depending on what you like. You also have the Metagenomics, which I'll talk about next. And we've been comparing a lot of different analysis tools, and ideally, we think we may end up having to do sort of a hybrid version of some. Some of them are much better than others. Our favorite right now is still Metaflan, even though it told us that there was plague and anthrax in the subway, but it's still, of all the tools we use, it's still the most conservative based on positive controls. And ideally, you should take this in an analysis where you have convergent taxa, you predict your ancestry to confirm who the patient is that you think it is, and then look for antibiotic resistance drivers is the goal. And you can even look for novel drugs that are present. So that leads to data integration, not only just within a hospital, We can also think of a larger scale. So this is something we're doing with customers to think about building a smart city. What if we're tracking everyone in the city, where people eat, essentially where they're getting their food, what's in their pets, what microbes do they pick up from their church or from their different transit methods. And so we started actually working this kind of a dream where instead of getting an alert about the child being abducted, you might say there's an alert for a pathogenic microbe detected. But I wouldn't even stop there. Let's say, for example, you compare sort of your immunological profile and see what is your known response to different potential antigens in the environment. Merge that with the map data, and actually you can get it maybe on your watch. So actually, if you buy an iWatch next week, you'll be able to download the ResearchKit app, and you can see all the path map data if you want to browse. It doesn't help you if you're not in New York City, but eventually we'll have other cities done. And those are just covered by their keynotes that you can actually do this and download the data. And so, in theory, in the future, which might not be tomorrow or next year, but sometime soon, as you can actually use all these data together to do comparative immunological analysis and say either that you're safe or it looks like you could use some antigen challenges, you should go there. And I won't say lick the polls, but I'll say you should just go to be exposed to antigens. And so, you can imagine a future where we're much more intelligent about where you go and how you go there. So, which leads obviously then to some technology questions. We've been doing a bit of making it faster, making it smaller. Some of the more recent work in the lab is to look at nanopore sequencing. So in this case, it's these really tiny sequencers that depend on alpha hemolysin. It's this beautiful octet that is about the same aperture of DNA. And you can order these. It's a beta program right now, but you can put an application, and usually they accept it. Except for one guy who's a competitor I know who didn't get one. But in any case, you can order them. They're this big, and you can plug them into a laptop and do sequencing. And it depends on the fact that as you essentially put a DNA through this pore, you have 5 bases at a time that are being measured. And so essentially you look for a hidden markup model to build what is the profile of DNA as it moves through the pore like this. So if it's 5 bases at a time, you assume ACGT, that's 1,024 possible states. And so they build this model and try to measure things that moves through the pore. This is what the raw data looks like right now, so it's not quite ready for prime time, I'd say. But what's amazing is it's gotten better, much faster than a lot of their technologies. It changes almost every 2 weeks. And now we can see about 71 kilobase reads, even at this point fairly routinely. Get very, very long reads, very, very fast, within a few hours. And sample prep is really the biggest bottleneck in speed. So it's kind of exciting time to think of a suitcase sequencer. You could walk in anywhere and sequence something as needed and move around to the next spot. So just to give you a sense of what the data looks like, though, is this is the raw data, this is the signal from the read. And so the goal is to try and merge these two together and do it really fast. And so I've done a little bit of thinking about this, like how fast could it get? Could it be like that scene in Gattaca where Uma Thurman, who keeps calling me, by the way, I gotta call her back. That's not true. She's not calling me. But Uma Thurman kisses Ethan Hawke. She's on a date with him. She's like, I don't know if I should date this guy. I'm gonna go get his genome sequence in this back room. And it only takes 2 minutes in the movie. And when this movie came out, everyone thought, well, there's no way. It takes billions of dollars to sequence the genome, and it takes years. That world is never going to happen. Because, by the way, he was using fake DNA, so he looked okay, and most of you probably know this movie. And then there was some drama, worked out fine. In the end, he went to space. So it was seen as impossible, but actually, it's in theory potentially doable on this technology if you can move something through the pore at 10 kb per second. Right now, they're near hundreds of kilobases per second of DNA moving through the pore. But if you make it a little bit faster and you have enough of them in one room and you have 30 grand to spend, you could get something down for 2.6 minutes, and that would be sort of pennies or a few dollars per gigabase of data. So it's not impossible. It just depends on this speed being fast and having enough of them in one room. And so I just did this calculation to see, is it even possible? And the answer is yes, actually. So you could imagine a time being in the subway where people are swabbing. There's a network where they're sequencing. There's no one who runs the machine a lot and just run around the subway and sequence things. So which was what we're kind of doing here. So the last thing I'll close on in the last 2 minutes is kind of the astronauts as a promise they get space. And so some people get upset when you say big data. We talk about big data. This is actually a meme generator. I actually printed this and put this up in the lab because people get really upset about the term big data or get a bit tired of it. But it's a problem I think we all face, actually, is that this is what my desk looked like for a while. So you got 50 terabytes of data sitting on the desk, or we're doing a large project with 1,000 human genomes, and I started just doing some math and thinking, well, let's say, okay, so we're going to do 1,000 genomes. It's about 100 gigs per genome. That's 100 terabytes of data. Let's say we did it for everyone that was in Russia next year, for example. It's 190 petabytes. That's a lot. But that's not that bad. We could figure out a way to manage it. But if you say you did everyone over for the U.S., that's actually 10 exabytes, 100 million genomes. If you did everyone in China, that's 130 exabytes. And let's just say we're in this future world where your genome gets sequenced every time you go to the doctor, and there's 10 billion genomes with 10 time points. That's 10 zettabytes. That's not that far away. It's not that hard to get there. And that's just the genome, right? That doesn't get at, say, the transcriptome, the epitranscriptome, the epigenome, and their interaction, gene regulation, nucleosome changes, or proteomic changes. They're looking at, say, metabolomic changes and their interactions, transcription factor binding sites, and mRNA degradation, or known annotations databases, kind of medical history, bacteria flowing out of their proteome, genome, transcriptome, and the viruses that are infecting them, plus drugs, vitamins, and hormones you have. And then you compare that to your environment, for example, do it over time. You could reach what's called a yottabyte of information, which is a real word. In this case, you could actually then have, you'd have to move everyone out of Delaware and Rhode Island to make space for the data centers if you wanted to store a yottabyte of information. Some people say yottabyte. I like yottabyte personally. And so if you think about this, let's say, say you just want to do this. I'm going to make a yottabyte of information because remember, according to Yotta, there's only two options. There's do or do not. There's no try. So if you want to just do it, you say, I'm going to make a yottabyte. How much is that? So you go online, say it's $100 per terabyte. You need 1 trillion terabytes per yottabyte. That's $100 trillion, which is, let's see, it's more than the money of the world's GDP, which is right here, is $100 million. So you couldn't even buy a yottabyte if you wanted to. There's all this data. And so then people say, well, maybe you just throw away the data. And I think that one of the big challenges is don't keep all the data, just keep the variants, or pick your data carefully, which we can't do yet because we don't even know which of the data I think is yet important. And the other challenge is that's just on Earth. Like, let's say you just go and you want to collect more data, and you have other telemetry data. Let's say you're now in space, and this is actually, I think, the biggest data challenge that we have in the lab right now is actually doing this sort of longitudinal integrative systems biology, one in space and one on Earth. So what we're doing is a project called the NASA Twins Project, which just got funded to plan a mission in Houston. It's basically going to be on the space station. So we have our it's part of the HRP, which is to study sort of understand how human body responds to space. NASA does still want to go to Mars by 2035. It's still on their plan. And you can see here, this is when we're supposed to start going to Mars. That by 2027 is phase A on their plans. These are all public slides. And we have to figure out essentially just different risk factors and understand have we gotten to an acceptable point of risk. And so one of them is to understand what happens to the body during long-term space travel. This is called the Twins Project. We're looking at two identical twins, one on Earth, one in space. Putting them up in the space station, collecting everything from stool to blood. You can see you have to collect their own blood and serography, which is not easy, by the way. And then stored in the space station and brought back down to Earth. And so we'll be looking at essentially genetic information, epigenetic information, both hydroxyl and DNA methylation, RNA expression, RNA methylation, proteomic changes, chromosome changes, antibody titers, B cell, T cell, telomere length, cytokines, targeted global metabolomics, microbiome, cognition, and vascular changes every 30 to 45 days for the next year, actually, because the launch just happened. So this, I think, is what is I think the exemplar of the big challenge. When we start to add all these data and longitudinal data and your sequencing genomes this much, where does the mutation here propagate through the system? How much does it matter to keep this data versus these data? Which ones are the most important? So I'm hoping that with this data, while it's a pilot data, so it's only 2 people, it's not built for statistical power, it's built for a data integration challenge. And I think this is our challenge today. So the launch just happened. Very excited about it, and at the end, I just want to thank. Well, actually, this is actually the official NASA poster. Here's the Death Star you can see there, and then this is part of our 500-year plan, which we're mostly on track for. So at the end, I'd say this is the lab. These people are fantastic. I want to thank collaborators, friends, who helped with the sampling and funding from the NIH and NRMDS, XSEED, and NASA, and the Sloan Foundation. And thank you for your time.
Questions?
So the cleaning crew reminded me of what I experienced when I was a postdoc at Yale that night. And back then I always had this feeling that they're not cleaning, they're distributing.
Right.
And so what would then predict that those folks that do that have the best immune system, right? Because they got the most distributed even exposure. Would you agree with that?
So in a priori, yes, that their exposure would be good, assuming they're healthy, otherwise healthy. But there's been no study yet of essentially those workers and their general health. I think it depends on what they're getting exposed to. Like in general, it's good to be exposed to more antigens, but it depends on what those are. And so their exposure to lots of other subterranean things or fungi or whatever is being carried by rats may not be as good as, say, walking through a park for a normal human. So there's questions like that also reminds me of, say, airline, basically people working in airlines have higher exposure to radiation, but they don't have a high risk of cancer. And so some theories that maybe having a low level exposure to higher radiation, at the very least, isn't deleterious, but maybe even is protective because your body's working harder for DNA repairs. And so maybe there's something like that happening, but there's no data, no epidemiological data, no study that's done that yet. So we're talking to the MT a lot about the Metropolitan Transit Authority about how to actually work with them more closely for things like that. But we don't know. Yes.
Your expression data on single cells was really interesting. So every cell has a different expression pattern. So say you're comparing, 1,000 cells of a control and 1,000 cells of a disease, say cancer. Would you see like a general trend in expression for those 1,000 cells that are going to, be useful diagnostically to see whether they're different from the control state?
Yes, so that's exactly what we're looking at before and after chemotherapy. And so far, the data I can present here, but we can see hematological development in terms of those, just the gene sets that are enriched, and essentially after therapy, if there is dysregulation, which should be hematopoiesis, which is from an AML sample. So it makes some sense pathway-wise of what's changing, but we can see what's interesting is that of the cells carrying that signature, it's only 60 or 70%. It's not as if all the cells carry that signature, and so there's a real heterogeneity. That even though you'll see some changes that make some degree of sense, it's not all the cells. And also, if you see a mutation in cells, there's a lot of monoallelic expression of those mutations as well as the transcripts. And so our big concern now is how much of that's technical. If you do single cell amplification of RNA, how much did you have accidental amplification of only one allele and not the other? So we're looking at spike-ins to check for that. But at least from the preliminary data, there's even more heterogeneity there, but you do see signatures in, say, 60, 70% of the cells that carry something that would look like it's responded therapy.
Thank you. I was a little disappointed to see the paucity of viruses on the cell line.
Oh, yes. So this could be to some degree our enrichment method, which was just the mobile. One thing that's in the paper is the top-ranking viruses were just phages, the bacteriophages. But a cool thing is that the top-ranking bacteriophages would often match the top-ranking bacteria. So it's a way to sort of cross-kingdom confirm what you think is there. But mostly it was just bacteriophages. Although in the supplementary tables, if you want to dive in, we didn't report this in the paper because most of the blast results we say, well, it's the top hit, but it's kind of a caveat emptor. But we'll see things like HPV. We saw herpes virus there. And basically one readout of a billion technically matched. The best match was to Ebola, for example. Of course, we didn't report that in the paper because one is an RNA virus, so we shouldn't even be seeing it. And two, which is it's one readout of over 10 billion. That was the closest match you could find. And so, to some degree, the big challenge that the paper's kind of created and even highlighted is that the metagenomics analysis field it's in pure infancy. So the databases are incomplete. We need more annotations. We need better tools. But there are some viruses there.
So, do you visualize in the future that animal models will be designed based on the microbiome of the specific target region, let's say gastrointestinal drug targeting, then you have engineered animals that have similar microbiome to, say, the human target?
Definitely. I think for as much as, I guess, in terms of personally, I view this as, for a little while, I was obsessed with all the novel non-coding RNAs that we kept discovering. A lot of my thesis work was on that. So in the last few years, I really shifted to thinking more about how there's this huge world of undiscovered microbes, sometimes already right on us and in us, that for sure are a component of health, or at least a response to drugs. And we just have started to look at them. So I think in the future, they'll be part of the design, they'll be part of at least the characterization. It'll be part of health. It'll be really integrated in terms of what's there. Definitely.
I think we need to move on, and we can talk to Chris more at lunch in the poster session. But the next talk is Dr. Kui. Thank you, Chris.